All right, so welcome everyone, and thank you for joining us for this ASEM's virtual visitor webinar. Today it's my pleasure to introduce Professor Ruth Baker, who is a Professor of Applied Mathematics at the Mathematical Institute, University of Oxford, a tutorial fellow in mathematics at St Hugh's College, and also a recipient of a Royal Society Wolfson Research Merit Award. Ruth leads the Quantitative Developmental Biology Group at the Wolfson Centre for Mathematical Biology, and her work focuses on the development and application of novel mathematical and computational methods and modelling frameworks for investigating developmental biology systems at the cell and tissue level. And the thing that I really like about Ruth's work is that it's very multidisciplinary, involves theoreticians, life scientists, and great international collaborations, including several people who are listening in at the moment. It's also been my pleasure to work with Ruth over the last 5 years as well. So I will hand it over to Ruth now to share her talk on quantifying the effect of electric fields on single cell motility.
Thanks, David, and thanks for the invitation. So just to say, almost as an apology up front, this is a brand new out of the box talk. So apologies if it's not quite as sort of polished as it could be. And on that note, if there's anything that's unclear or anything you think I forgot to say, please do just shout out. I can't see the chat, but I'm more than happy for sort of people to ask for clarification on anything. And the other one is I hope my voice gets through to the end of this. It's a bit of a cold over the last few days. Okay, so this is a project all about understanding how electric fields impact single cell motility, but I thought that because I don't know sort of everyone in the audience, I'd tell you a little bit about what we sort of try and do in the group more generally. So our focus is mostly understanding mechanisms that drive collective cell motility and how they combine with cell adhesion, proliferation, and cell death to contribute to very complicated biological processes. And we mostly focus, as David said, on aspects of embryonic development, but also thinking more about how understanding aspects of embryonic development help us to understand pathologies, diseases such as tumor growth, and also repair and tissue regeneration. And more increasingly, I guess, our goal is to take mathematical models or to develop those models and calibrate them to data so that we can really interrogate sort of biological mechanisms. And like probably many people in this audience, we try to take a multidisciplinary or an interdisciplinary methodology much of the time. So we collaborate with experimental groups who are looking both in wild type and perturbed scenarios to understand a particular biological question. And then we develop the mathematical modeling frameworks, the simulations of those models, and then develop and use means for data analysis and model testing to connect the experimental data in the models. And we're thinking much of the time about this sort of cycle of predict, test, refine, and predict, where essentially we might have some predictions, for example, coming from some experiments, so experimental hypotheses. And we want to integrate those hypotheses into a mathematical model so that we can test them. And often as not, we learn much more when we get something slightly wrong about those hypotheses. There's something about the model predictions that aren't consistent with the data, and that enables us to go away to refine our hypotheses, make some experimentally testable predictions, and then the cycle sort of begins again. So what I would say is that today I'm going to talk about something. What I'm going to talk about is going to get almost to sort of closing that loop. But there's much more work to be done in this area. Okay, so thinking about sort of directed cell migration more generally, then what we do know is that the cells have the ability to respond to a huge variety of types of environmental cues. So the one in the movie is sort of cell leukocytes undergoing chemotaxis, and these are cells from mouse. And essentially what's happening here is that there's a diffusible chemical in the environment. The chemical diffuses from a localized source, it decays or is taken up by the cells, and this creates a gradient in the chemical. And so chemotaxis is all about cells undergoing directed migration up generally gradients in chemical concentration. And that's, I guess that would be positive chemotaxis, and then the reverse being negative would be motion down gradients of chemical concentration. So chemotaxis is maybe one of the best known and quite well studied, but we've got many other types of sort of taxis, if you like. So haptotaxis being substrate bound or migration up gradients of substrate bound chemicals or adhesion sites. Durotaxis being migration up rigidity gradients or gradients in extracellular matrix stiffness. So these are all really kind of fairly well studied mechanisms by which we can get directed cell migration. But one that's talked about a little bit less is electrotaxis. So this is the directed motion of cells due to an applied electric field. So you can see here in this movie, for the first portion of the movie, the field is kind of left to right and all the cells shift in that direction. And as we shift the field then to up down, or switch the field from left right to up down, you can see the cells kind of turn the corner and they go in an upwards direction. So it's known that a wide variety of cells can actually sense and follow DC electric fields, such as this type of cell. So these are MDCK cells, I think. And we also know that these kinds of fields arise naturally in biological tissues, both during development and during, for example, wound repair. What I really like about Electrotaxis is it's hugely controllable. So essentially we can design we can put electrodes into these into a dish, and we can precisely design an electric field stimulation pattern to get a desired output migratory pattern. Or at least in principle we can do that. But there's a huge amount that we still don't understand essentially about this process of electrotaxis that I think needs to be really sort of unpicked before we're able to use electrotaxis as a controller. And this is just another example with a different cell type. These are human corneal epithelial cells, and you can see that in this movie, essentially for the first portion of the movie, there's no field switched on, so it's here. And then you can see when the field is switched on, all these cells are beginning to move up to the top of the image, and then the field direction is reversed, and they change their mind and they go follow the field again towards the top. So yeah, so I'm very interested in sort of better understanding this process and thinking about how then to potentially develop sort of new therapeutic options. So for example, in chronic wound healing. And actually, we're already beginning to see people try to use electrocurrents to essentially accelerate wound healing. So there's this notion of a bioelectric plaster. And this is, I'm not convinced that these experiments totally show that these are effective, but the idea is that you have essentially a plaster that has some anodes and cathodes embedded in, and there you can essentially put a current across a wound. And the idea is that somehow that, and we don't really understand how, that accelerates sort of wound healing, at least in these mice. So what I really want to do is to use mathematical models to really better understand how these electric fields are impacting cell migration. Having said that, I'm not going to focus on the real the details of what's happening in terms of their subcellular sort of signal transduction and the events that lead to cell motility. What really interesting in this project was understanding essentially what aspects of cell motility were impacted by the field, and then how we can therefore turn that knowledge into being able to design sort of control sequences, or being able to predict what cells will do under an applied electric field. Yeah, so as I said, kind of the overarching goal, can we turn Electrotaxis into a cellular sort of bioengineering tool? But to start with, can we just really understand what single cells are doing? So this sort of, I think, towards being sort of to get to the target of being able to understand how to control a collective of cells, are sort of, I think, some baby steps you need to take along the way. So we already kind of have, I guess, a relatively good understanding and numerous models for how single cells behave in terms of their motility in the absence of an electric field. And that has been nicely scaled up by many different people to think about the behavior of cellular collectives. So what does the motile behavior of a population of cells look like? So the aim that we want to take sort of in this talk is to think about how single cells behave under an applied DC electric field. with the aim being that eventually we can sort of put all that knowledge together to think about what happens to cellular collectives under an applied electric field. But as I said today, excuse me, for today it's going to be enough to talk about what happens with sort of single cell motility. Okay, so what I first wanted to show you is the data that we're going to be using in this experiment. So these are movies of the motility of human corneal epithelial cells. So we've got 5 hours worth of movie and imaged at 5-minute intervals. So in each case, we've got two data sets that we're going to use. So data set 1, data set 2. And this is just control data. So there's no electric field applied here. We're just going to see that cells are essentially spontaneously polarizing and moving randomly. Now, one thing I should say at the outset is that we're assuming we're going to build some models that are of single cell motility, and we're going to ignore cell-cell interactions. And essentially, there are obviously clearly cell-cell interactions in these experiments, but we're going to hope that the impact of them is low enough that we can really still be able to characterize sort of the single cell behavior. Go on, everyone have a question? Shout out if you do. No. Okay, so this is the control data that we're going to use. And effectively what we're going to do is track the positions of up to about 30 cells from each of these movies over the duration of the 5-hour movie. And then we've also got 2 data sets from some similar experiments, but ones in which in fact an electric field was applied. So what's going on in here, same cell type, same image frequency over the sort of the 5 hours. For the first hour of this experiment, then the cells you can see here were essentially moving just randomly without an applied electric field. And then for hours 2 and 3, you can see that there's a field applied from left to right, and they all start moving from left to right. And then from hours 4 to 5, then the field direction was reversed, and so they all start moving from right to left. So you can see there's quite a nice switch in their behavior as that happens. What I'm going to do, though, is I'm going to just use a portion of this experiment to calibrate the data, and I'm going to reserve a portion of this experiment in order to test the model. So effectively, I'm going to use just hours 1, 2, and 3 of this experiment to calibrate a model of single cell electrotaxis, and I'm going to reserve the data from hours 4 and 5 to test the predictions of that calibrated model. Okay, so first of all, I just want to talk a little bit about what we're going to call the autonomous model and how we calibrate it. So this is going to be a very, very simple model of cell motility in the absence of an electric field. So it's very, very simple in the sense that we're just thinking about position X changing as a result of the cell having some velocity V at time t. And then essentially V in the absence of electric field is just V cell, that's just notational. But the model actually all drills down to the fact that we're going to assume that we can write the velocity of the cell in terms of a scalar V, that just being the sort of modal cell speed when it's polarized, and then a vector P, which represents the cell polarity. And so this whole model comes down to really thinking about how the cell polarity evolves over time. So this is just a simple SDE that tells us how cell polarity evolves. And the thing to note really here is that, excuse me, is that cell polarity is or the evolution of cell polarity is really driven by this potential function W. So this was really designed to be a very, very simple model of cell motility that just captures the really essential aspects that we could see in the data. So when it came to thinking about how to formulate W, then the idea was really to go and to eyeball the data we have from the sort of the control experiments. So here I'm just showing you, at least in the top 2 figures, this is the distribution of displacement distances traveled by cells in each 5-minute sampling interval. So there's one line here for each, or one curve here for each of the cells. And so they're all smooth just using a kernel density estimate. So essentially, really, they're quite fairly consistent with the assumption that cells can spontaneously polarize, and in which when they have polarized, they travel with some positive speed, and that they can also transiently depolarize. And if you look at the data, you can sort of see that just eyeballing the movies as well. And then the sort of autocorrelation plots, which you see below that, just tell you that the kind of autocorrelation in the observed motility direction just decays essentially gradually over time, sort of consistent with the fact that the velocity direction or the polarity is going to evolve according to a random walk. So essentially, in terms of the model for the evolution of cell polarity, what we're essentially seeing in this control movies is that cells can spontaneously polarize, and that they're, in the absence of a field, their subsequent direction of motion is unbiased. So we're just going to get, let W be a function of the scalar P, where P is the absolute value of the vector P, and then P is just this function down here at the bottom. Okay. So the idea really then is, oh, hang on. Yeah, that's on the side. Yeah, so the idea really is that P stochastically evolves essentially in their regime where mod P is approximately 1. So cells essentially are generally sort of polarized, but the direction in which they're sort of polarized is uniformly distributed on 0 to pi. And then they can transiently depolarize. So the autonomous model for cell motility just looks like this. So essentially, really what you're doing is simulating how the polarity evolves over time, and then you can then just integrate up to get the evolution of the position of a cell over time. So we have one of these models for every cell that we track. And then what's important to note, I guess the rate at which cells sort of transiently depolarize is determined by two parameters in the model. The first is the timescale parameter D, and the other one is, so you can either think of it in terms of this parameter beta, or what I'm going to do is actually think of it in terms of a potential energy barrier, which is related, so that delta W is just related to beta over 4. So the idea being then that we've got essentially 3 free parameters in this model. We're thinking about this B, or in fact this scalar value, the modal value of so essentially the cell speed. And we're thinking about the timescale parameter D, and we're thinking about the potential energy barrier delta W when it comes to calibrating the model to data. So the first thing that we did was to then essentially take this model and attempt to infer the parameters V, D, and delta W using the tracks that we collected from those movies that you just saw. So we're going to take a Bayesian approach to model calibration. So we're thinking about the posterior distribution of parameters theta, and this is given data collected in the absence of an electric field. And then we get at that in terms of evaluating the likelihood of the data in the absence of the field given parameters theta. And then the prior distribution just represents any previous knowledge that we might have about the parameters in the model. And so just to highlight again, the parameters of the model are the cell speed, or the modal cell speed, the timescale constant, and the energy barrier delta W. And we just essentially think of this in terms of the product of the likelihoods of each of those individual cell tracks. So the likelihoods intractable, so we needed to use something that was likelihood free, and we went down the route of using a synthetic Bayes approach. So that requires us to do a couple of things. The first is to define some of the summary statistics of the trajectories, and I can show you those later if it's of interest, but there's many that you can take, but we settled on a group of 4 summary statistics. And then once we've done that, we're just going to approximate the likelihood with the synthetic likelihood, essentially the likelihood of the summarized data under the fitted empirical Gaussian distribution. And I know lots of people at QUT work on this, so I'm going to assume for now that you're fairly au fait with all of these methods. But I just wanted to say that we're essentially going to use synthetic Bayes to estimate the model parameters. And here's the results of doing that. So on the main diagonal, I've got the marginal posterior distributions for each of the parameters V, delta W, and D. And then on the sort of below that, in the lower triangular bit, I've got the bivariate posterior distributions. Go on, does someone have a question? I've got the bivariate posterior distributions, and they're just indicated or drawn over the full support of the prior. So you can see that essentially we started with a fairly wide prior because we didn't have much knowledge about many of these parameters previously, especially I guess I would say sort of delta W and D. But you can see that we can really kind of sort of narrow the support of the posterior down into quite a small region of that sort of prior space. And then on the upper sort of triangle, I've just zoomed in on those posteriors a bit so you can really sort of see what they look like. But I think the take-home message is that without putting any real strong assumptions on the values that these parameters should take, we can get fairly good and fairly tight estimates of each of them, which is nice. And then going forward from there, we wanted to do a posterior predictive check for this model. So I guess there's a few different things that we could do really. So the first thing to do is that we check that if we just infer the parameters sort of using each one of those sort of data sets independently, so replicate 1 or replicate 2, or we infer the parameters using all of the data together, that the posteriors don't look terribly different to one another. And you can see there's a few differences when it comes to replicate 1, but I think overall they're fairly consistent across the data sets, which is really nice. And then the other thing we did was a posterior predictive check, sampling essentially parameters from the estimated posterior distributions and simulating the model, and then looking at how the distribution of outputs from the model were consistent with the data. So the data in these plots, the data are just shown in these sort of little vertical lines, and then the solid lines just show the outputs from the model predictions. And what I'm doing here is just comparing for each of the 4 summary statistics that we used in the synthetic Bayes approach, just comparing the outputs between the model, or the predictions of the model with the data. And I think generally they look sort of pretty good. And then the other thing that you can do is you can also just check how, do a bit of a visual check, I guess, as to how the sort of observed position. So these are essentially the tracks that we get from the data, all translated to the origin, check sort of visually what they look like compared to the simulated positions that would come out of the model. And again, this is sampling from the posterior distribution repeatedly in order to, and then simulating the model. And I don't think it's too bad. I mean, I think sort of visually you can see sort of differences in the two sets of trajectories, but probably we're doing a reasonable job. And maybe we can talk more about this later, because I'm definitely not an expert in synthetic Bayes, but I think there's probably a better agreement with the summary statistics, which is maybe what you'd expect. And there's certainly some loss of information when it comes to thinking about then simulating the actual tracks themselves. And I guess the other thing I wanted to say that we can do with this model is make some predictions over cell characteristics that are sort of hard or impossible to measure. So we can do things like look at the distributions of times to polarization or depolarization, or the probability that by the end of the experiment, a cell is polarized. And so there's some quite nice predictions that come out of that, which we sort of can't get at from the data. Okay, so by this point, what's nice is that we have a model that's relatively good at predicting the sort of behavior of cells, these cells, so they're human corneal epithelial cells, relatively good at predicting what they do in the absence of an electric field. And then we wanted to turn it around and say, okay, what happens when we apply an electric field? So the electric field we have is a 200 millivolt per millimeter field, and it's applied in the positive X direction. And in the movies that you saw, essentially, and this should be minutes really, but so with the electric field, there's no field at all for the first 60 minutes of the experiment. And then for between minutes 60 and 180, the field is directed from left to right. So the cells want to go from left to right, and then it's reversed for the final 2 hours of the experiment. And we're just asking the question, how does this applied electric field impact cell motility? And so we're just kind of asking the question sort of mechanistically at the level at which we're modeling the cells, what's going on and where's the impact of cell motility being seen? And just to remind you, we have this very simple model where essentially we're just looking at how the polarity of a cell evolves over time. and everything really depends on the parameters v, d, and beta or delta w, which was all wrapped up in this potential function w. So what was the idea here? The idea was to really come up with, just talking with sort of collaborators in and amongst the group, just to come up with a range of different ways by which we think that applying electric fields to these cell populations might impact their cell motility. And these are kind of the four different potential ways that made the cut. So the first is to assume that if you apply an electric field, there's some kind of velocity bias. So essentially, v of t is the velocity of cell, is the cell velocity, and then we can write this in both in terms of the velocity of the cell that we would have, and this is the kind of autonomous velocity in the absence of an electric field, plus an additional component. So we're sort of assuming that the electric field imparts a force on the cell, which changes its velocity directly. And then essentially we're going to say that this is this velocity is directed, it's in the same direction as the electric field, and that it's basically essentially gamma 1 times the modal cell velocity. So we're thinking of gamma 1 as being a relatively small parameter that perturbs the electric field, that perturbs the motility of the cell in the direction of the electric field. And so I guess the sort of strength of this is measured in terms of its velocity in the absence of an electric field. That makes sense, hopefully. So number two, the second way in which we hypothesized that the electric field could be impacting the motility of the cell was by a speed increase. So if we take the sort of take the original model and think about the velocity of the cell, what we're saying is that before it was just v times the polarity, and now we're saying that there's an additional component. So it's going to increase the speed of the cell by a sort of percentage or a fraction gamma 2 times essentially the strength of the field. So it's not having an impact on the direction in which the cell's moving, but it's just going to make the cell move a bit quicker as a result of there being an electric field. The other one, the next one, was a speed alignment hypothesis. So it really just says that it will sort of impact the cell based on how aligned the polarity of the cell is with the electric field. So p hat here is just a unit vector in the direction of the polarity of the cell. And we're just saying that essentially how aligned the cell polarity is with the electric field determines what the essentially the increase in speed is. Okay, so these two are sort of can be thought of as quite similar impact. And then the final way which we thought that that the electric field could impact the behaviour of the cell was in terms of imparting a polarity bias. So the first three mechanisms all impacted sort of the velocity of the cell directly, whereas in the last mechanism we're going to go in and we're going to play around with the polarity of the cell. And essentially we're just going to say that the polarity is biased in the direction of the electric field. Okay. So we had a range of different sort of ways in which we'd hypothesized that essentially I'm trying to say that the electric field could actually bias the cell motility. And it's quite, I guess this diagram I think is quite useful because it provides a means to think, at least for three of them, about how they would impact the sort of the polarity of the cell, or the velocity of the cell, if you like, at stationarity. So under the autonomous model, what we're seeing essentially is that the cell wants to more or less sit with a speed of v, so that's the modal cell speed, and its polarity is uniformly distributed, so it's moving uniformly in any particular direction. It doesn't have any kind of bias to its velocity over the unit circle. And then if you move into a model that includes these different electrotactic mechanisms, you can see how you sort of shift that velocity. So if the electric field was applied from sort of left to right, you can see that the impact of the speed increase mechanism is to just essentially increase the radius of that potential sort of where the cell wants to sit. So there's an increased radius, if you like. So we're just increasing essentially the speed of the cell by a factor gamma 2. Gamma 1 was this idea that the electric field would essentially just impart an additional force on the cell in the direction of the field. So that's just translated this profile to the right a little bit. And then gamma 3 sort of just skews the velocity distribution, because it just says, okay, if a cell polarity is aligned with the electric field, so that would be essentially if the cell is moving, the cell is sitting around here, it's on the axis and it's moving in the positive x direction, then I see the greatest increase in essentially in velocity. So that parameter gamma 3 just skews this distribution. So that's how essentially the distribution of velocities would change as we put in these different electrotactic effects. Okay, so we've got a set of 4 different electrotactic effects, and what we really wanted to do was to try and use the data to understand which one of them or which combination of them might be at play and be actually impacting sort of cell motility. So we went back, and now we have a sort of another calibration problem to do. So this time, going to take the same approach, but this time we've got essentially lots more parameters in the model. So we've got the original 3 parameters of the autonomous model, and then we've got these additional sort of 4 parameters, gamma 1 to gamma 4, which control each one of these different hypothetical mechanisms. So we're going to take a Bayesian approach to parameter inference again, where we now include the data that comes from the control scenario, so where there's no electric field, and the data that comes from the experiments with a field. And we're going to access the posterior distribution by thinking essentially through the likelihood of observing these data given parameters theta. And just to remind you that the data is both the data without an applied electric field and the data that had an applied electric field right up until the electric field direction was reversed. So in terms of these movies, essentially, we're just looking at the first 3 hours of these movies in order to calibrate the model to the data. Okay. So first of all, we really wanted to kind of get at the question, which electrotactic effect? So what we're going to do is we're going to think of those 4 different potential mechanisms as giving rise to a collection of 16 different potential models. So essentially all combinations of the possible different electrotactic effects. So it could just be 1, 2, 3, or 4 on its own, or it could be any combination thereof. Excuse me. And we're going to essentially look at the objective function J mu as characterizing how well each model exits the data. So when I think of model X, it's basically just saying which combination of these 4 possible mechanisms gives rise to the data or is included in the model. And then we're going to, in terms of mu, there's 2 contributions. In terms of J, sorry, there's 2 contributions. The first is essentially represents the sort of partition function or the probability of model X, giving rise to the data both with and without an electric field. And so that's just the integral of the likelihood over the prior distribution multiplied by the prior distribution for that model. And then what we do within our objective function as well is we essentially include a regularization term that sort of penalizes models that have more parameters or mechanisms in them, right? So we have a regularization parameter mu, and then this term in here. So the 3 just comes from the fact that the original autonomous model, in the absence of an electric field, already had 3 parameters in it. And then mod X is essentially just tells you how many of these hypothesized electrotactic effects are included into the model. And if we take mu equals 2, then that essentially corresponds to the Akaki information criterion. Okay, so let's look at what happens when we when we do this sort of model result with this model comparison. So for this data set, at least, excuse me, what we're seeing is that effectively it's mechanism 4, so this is just including mechanism 4 in the model, gives us the best fit of the model essentially to the data. And that's the case essentially whether we include regularization or or not. So this is without regularization, and this was with regularization. And you can see that, and actually the sort of next three most favored models, at least with regularization, were those that included the combinations of the other mechanisms with mechanism 4, but certainly that was the sort of most important mechanism to include in the model. So to summarize, what I've said here really is that I think there's quite strong support for the hypothesis that the bias we see in cell motility as a result of this electric field being applied comes sort of solely through cells preferentially polarizing in the direction of the electric field. So this is essentially the model where we modulate the evolution of the polarity by including this extra term with strength gamma 4. Okay, so and then once the other thing that once you get there, the other thing that you can do is then go away and you can look at the posterior distributions that come out of just that model, which it solely includes the polarity bias as an electrotactic effect upon the cell motility. And once again, I think what you can see, so these are the marginal posteriors along the diagonal, is that we get reasonably confident sort of estimates. All of the parameters in the model are identifiable, I think, given the data. And although we start from a fairly wide sort of or broad prior distribution, we can really narrow into a quite small region of the parameter space in terms of where the mass of the posterior actually sits, which is nice. And again, I've provided sort of zoomed in profiles of the posterior distribution in the upper triangle. So from there, I guess the things that are sort of left to do are to do a posterior predictive check. So just to check that essentially the predictions that come from simulating the model using parameters randomly drawn from the posterior distribution are sort of sensible. And you can certainly see that in terms of the summary statistics of the synthetic Bayes approach, that seems to be the case, which is nice. And you can also compare the trajectories that you would sort of predict under both the the model without an applied electric field and that with an electric field, and compare them to data. And again, I think it's doing a reasonably good job. There's perhaps a bit of loss of information in moving from the summary statistics to the actual trajectories themselves, but I don't think it's bad. And then the final thing that we wanted to do, which is moving towards being able to close the loop here, is to make some predictions for that test data. So this is essentially what happens in the data in the last 2 hours of the movie. So we've kind of got an hour of an experiment without an electric field, and then we switch on the electric field to make the cells move towards the right for 2 hours. And then the final 2 hours that we haven't used is what happens when we switch the direction of the electric field, so that cells then start moving towards the left. And so we're going to use this as test data. So again, we can draw parameter values from the posterior distribution and use those to simulate the model and compare essentially the data, which is shown here in the black lines, with essentially the outcome of multiple model simulations. And you can see, I think, at least for the summary statistics that we used, that there's pretty good predictions made by the model in terms of the data. And then when we go and look at the actual trajectories that might come out of this, I think things are reasonable, actually. So these are the observed data. So again, what we've done is essentially taken the position of each of our tracked cells at the 3 hour mark, translated them to the origin, and looked at the trajectory of the cell as it moves over the subsequent 2 hours, or the remaining 2 hours of that experiment. And you can see that cells certainly turn around pretty quickly, and they begin moving to the left. And I think the main difference in our model is that cells take a little bit longer to sort of turn around, and the trajectories are a bit smoother. And once again, I could say that might be due to the use of the different summary statistics. But the other thing I was going to say is that I think that also could be down to the fact that we've actually kind of assumed that this data come from single cells, whereas in reality there's actually the cells plated at relatively low density. So there are some cell-cell interactions, and it could be at this point that those cell-cell interactions become a little bit more important in terms of the reorientation of cells. Okay, so I'm more or less done. I've got a couple more things, just one more thing I wanted to say after this, but sort of in terms of the biological conclusions, our results predict that for this cell type, so for human corneal epithelial cells, moving in this particular electric field, the bias in cell motility is coming only from cells wanting to preferentially polarize in the direction of the electric field. And I think one of the next steps for this particular cell type in this model is to then think about how the strength of the field relates to the bias and the motility we see. So the obvious thing to do is to repeat this kind of procedure using data collected using different field strengths to kind of build up a picture of how these parameters gamma 1 to gamma 4 might depend on the strength of the field. So it might be that we see different effects coming in at different field strengths, or it might just be that we only see sort of this sort of preferential sort of polarization, but that we can build up a picture of how gamma 4 relates to the sort of strength of the field. And I think that would be something that would be, hopefully we'll get onto over the next few months. But I should say that I think this framework is very easy to apply to other experimental scenarios and to other cell types. So I think it'd be very interesting to take a range of different cell types and just begin to understand whether for different cell types we kind of see the impact of the electric field presenting in the same way in terms of the mechanistic impact upon cell motility. And one of the reasons I think that is important and is a very interesting question is actually kind of a priori. When you take a new cell type and you put it into an electric field, we've got no means at the moment of predicting whether it's going to migrate towards the anode or towards the cathode, and actually it could be either, and we have no idea why that is. So I think it's perfectly reasonable to expect that we could pull out different mechanisms with different cell types, but this model allows you to do that very easily. And then obviously in the future, we'd really like to sort of extend this up to model populations of cells. And I think there's interesting questions surrounding whether you can sort of bolt on a model of cells, of how cells interact with one another, with a model of how cells react to the electric field, and sort of whether that will be sufficient to be able to model how cellular collectives move in an electric field, or whether there's going to be a feedback between the two. So do cells interact with one another differently because of the presence of the field or not? And I think the real goal of this is to develop optimal stimulation protocols, so that if we have a desired sort of cell trajectory, we'll be able to design a stimulation protocol that realizes that as closely as possible, and eventually to embed what we're doing into the actual sort of hardware and software that's actually generating those stimulation protocols. The final thing I wanted to say, just because I know there's a little bit of interest in, or lots of people working on synthetic Bayes at QUT, was that what we actually did under the hood was use sort of sequential Monte Carlo with synthetic likelihoods to target these posterior distributions. And that's really because I guess we didn't have much idea in advance of what some of the parameters of this model would look like, or any real idea about how big or how small they might be. So we wanted to take really quite broad Fourier distributions. So doing a kind of rejection sampling approach was effectively just going to be infeasible, particularly because we also needed to simulate the model multiple times. So the idea was that instead of doing sort of one round of sort of synthetic Bayes, we would put synthetic Bayes into a SMC algorithm, so that each intermediate distribution we are going to target a tempered distribution. So T essentially representing sort of temperature. So T is essentially going to be evolved over the sequence of intermediate distributions from 0 to 1. So N in here just represents the number of simulations we did of the model with each parameter value. So that was 500, I think. And what I was going to say is that at each generation, we use the effective sample size to adaptively increment the temperature. And then the final stage, we sort of post-process the final posterior to improve the sample quality. And that allowed us to essentially get a handle on these posterior distributions in a relatively okay sort of time frame computationally. So yeah, just to finish up there, I just wanted to acknowledge the people who were involved in this work. So all the theoretical sort of modeling and inference was done by Tom Prescott when he's a postdoc in my group. He left at the end of last week and he's about to take up another postdoc position at the Turing Institute. And this is all work done in collaboration with Min Zhao and Kan Zhu at UC Davis. So they did all the experiments and provided all the bio expertise. So with that, I'm going to finish and I'm happy to take any questions. Thanks for your attention.
Thanks very much, Ruth, for a really nice talk. Could everyone give a round of applause for Ruth, virtually or in real life? And have we got any questions for Ruth? I won't start them off. I have some listed, but I'll give others the opportunity to ask a question first. Just raise your hand or unmute is perfectly fine. Okay, I'll start then. I will start. So I was going to ask about the choice of synthetic likelihood. So synthetic likelihood is a pretty interesting technique, and there's certainly a lot of work done here at QUT on that technique. But what was the motivation for synthetic Bayes versus something like approximate Bayesian computation or similar? I know your group's done a lot of work in those areas as well. Could you?
Yeah, it's a good question.
What the choice was?
I guess I think probably Tom sort of wanted to try it out and see how well it works. Is probably one comment that's probably certainly true. But I think that we thought about using ABC because clearly we've used it a lot. It wasn't something where I guess ABC looked like it was going to take, certainly because we had sort of 16 models to compare, then ABC was going to be quite heavy computationally. And I think we felt that sort of having access to the synthetic likelihood was going to be something that would probably work in this situation. Like certainly if we eyeballed the sort of distribution of summary statistics, they looked like they would fit into that framework. They looked approximately normal. And we sort of just went from there really. I don't think it was particularly, I think it probably could have gone either way, but I think this is probably a bit more efficient for us at the end of the day.
Certainly the results look like it works pretty well.
Yeah. I think one of the things to say is a bit buried in this was the fact that, but you have this with ABC as well, unless you use some of the more semi-automatic methods, is that we struggled a little bit with thinking about what sensible summary statistics were. So if anyone has any thoughts on that, it'd be really good to hear. So certainly the summary statistics that seem to work best seem to depend a little bit on the cell type and the particular data set that you have. And it's a bit of kind of trial and error in a way to try and work out what summary statistics are informative when thinking about the posterior distribution. So it required a bit of tinkering with to get it to work in that sense.
Right, thanks a lot for that answer. We do have a question in the chat from Kai. Kai says, thank you for your talk. I have two questions, and if it's okay, could you post them to me? What do polarized cells mean biologically? Do they align along or perpendicular to field lines, or does the cytoskeleton preferentially form in one direction or something else? And then would the results have changed if you included a time lag in your model? Those cells probably need some time to react to an electric field.
Yeah, good question. So polarized biologically means that I guess effectively, depending on cell types, sort of cell needs to assemble the right kinds of sort of machinery from some biological or physical perspective into the right place to be able to migrate. So once it's assembled that machinery in terms of the sort of cytoskeleton, then it's sort of able to move in a particular direction. But if it wants to turn around or move in a different direction, then there's kind of some effort, if you like, involved in doing that, because it has to reassemble or sort of change its cytoskeleton. So in terms of from a biological perspective, it is sort of rearrangements in terms of internal structure of the cell. And would the results have changed if you included a time lag in your model? So yeah, this is something we thought about actually quite a lot, and it's a really good question. So I think there's two things in terms of the time lag. One is the fact that because cells are polarized, they do need time to sort of change their internal structure, and that's important to include. And there was obviously some kind of signal transduction pathway. So one of the things that I think, one of the motivations for actually going in and modeling everything at the level of the cell polarity was that then that takes sort of some time, if you like, to feed up into changes in the cell's sort of position, right? So there is an effective sort of time lag in there. But yeah, you're quite right that one could introduce an additional time lag that basically just says that there's, sorry, I don't know if you can hear like next door dog barking all the time. I'm trying to close the window, but what you could do is just include a time lag that just sort of says a cell is kind of what a cell is doing is a function of what the electric field was some time lag tau ago, and obviously that would have an impact as well. I don't think the latter of those two is probably super important, but I think this idea that the changes in the polarities to say sort of take some time to be realized in terms of changes in the position of the cells is more important. And I think what you can see in some of these, where has it gone? These posterior predictive checks I've showed you, wherever it was. Sorry, it appears on the slide somewhere. I can't see it now, is that what we saw is that when cells turned around, it took them a little bit of time in order to be able to turn around as precisely that behavior sort of coming through. And it's to do with the fact that in our model, the polarity is walking sort of randomly around the rim, if you like, of that potential function. And the rate at which it kind of walks randomly there is related to the timescale parameter D, and that's what sort of controls the lag, if that makes sense, hopefully.
Okay, Kai says thanks. Any other questions? We've still got 5 more minutes to talk. So if anyone, there must be some people who have insights into synthetic Bayes here and tuning synthetic Bayes. Philip has a question.
Yes. So on, I think it was slide 21, you show these velocity maps, how the expected velocity looks like. Yes, exactly. So why does the field in X direction changes the velocity in the Y direction?
Yeah, because effectively the gamma 2 parameter was just sort of saying that the fact that you've got an electric field, and it goes back up to here, the fact that you've got so the speed increase parameter was kind of agnostic of the field direction, right? So it just says you've got an increase in the cell speed by a factor of gamma 2 in any direction. That was just kind of hypothesized kind of in the model. So gamma 2 just kind of changes the cell speed sort of no matter what the field, where the field is directed. Whereas the speed alignment parameter just says that if you're polarized in the direction of the field, you increase your speed, if you like. So gamma 2 parameter is just increasing the cell speed generally, which is why the radius of the circle increases, whereas gamma 3, because it's polarized, so if you're sitting on this axis, in the positive part of this axis, gamma 3 is you're aligned with the field, so you see the greatest speed increase. And conversely, if you're kind of totally your polarity is totally opposite to the field, then your sort of your speed is decreased. So it was just a hypothesis, really. Sorry, something to test, but as we saw, it wasn't sort of the results of the analysis didn't suggest that that was something that was actually involved.
Okay, thank you.
Okay, we probably have time for one more question, if there's any more outstanding questions from the audience. Okay, Ruth, I was just wondering, the particular cell line that you're looking at, are there immediate applications for that cell line that you're thinking about for this?
Yeah, good question, David. So I guess the reason that I was using it as Min Zhao, our collaborator at UC Davis, is in the Department of Ophthalmology, so they're all about eyes, and so I guess that's why. We've thought about when you write these sort of pie in the sky sort of grant applications that need some very, very adventurous thinking. We suggested that we might be able to develop eventually contact lenses that can help be placed in the eye to help with things like wound healing. And so I guess there's kind of nice applications at that level. So kind of like a bioelectric plaster, but a contact lens version of that, I think could be sort of super cool. But I think, you know, that's a long way off. But the reason it is to do with the sort of focus of the collaborators, really.
Thanks.
We've got other collaborators at Princeton who are fairly agnostic probably to sort of cell types, so they use things like MDCK cells a lot, but I think it's quite nice to think that there could be applications of this eventually.
Yeah, definitely. I mean, I think even though you're saying it's pie in the sky, it's nice to think that these methods somewhere down the line have a positive impact.
I think so, it'd be great. I mean, I'm not, you know, the results I showed you from the bioelectric plaster experiments, I'm not sure I'm convinced about, because I think the hydrogel makes much more difference than the plaster itself. But certainly I think, you know, there are electric fields, endogenous electric fields involved in wound healing. And so if we can understand how to exploit those in order to accelerate wound healing, that could be a very, very powerful tool.
Thank you very much. So that brings us to the end of the hour. We should thank Ruth again for waking up very early in the morning to deliver her talk. So let's thank Ruth again. And I look forward to seeing everyone again in another talk. But again, thanks, Ruth, for your time. It's been great to also testing your presentation on us.
Thanks, yeah, thanks for letting me.
It certainly didn't look like it was a brand new presentation. So thank you very much for your time, and thanks everyone.
Thanks, David. Thanks, everyone. Bye.